think so.
Oh, there we go. Hello. Hi, James.
Hi.
It's nice to see you. I'm Angie Mikhail, and I'm a program officer at the Kavli Foundation. And today we are going to have a live chat with Dr. James Lee, who is a postdoctoral fellow in Corey Bardman's lab at the Rockefeller University. And just to provide a little bit of context, James was one of the Kavli Grass Fellows this past year, where he studied neural bases of arsenic tolerance. So, James, we're really excited to have you here. Thank you for joining us. And yeah, could you tell us a little bit about your work?
Definitely. Yeah. Thank you so much for having me. So I am a bit of a amateur nematologist. My scientific background is I did my PhD back in Caltech in the laboratory of Paul Sternberg. And ever since then, including currently as a postdoc in Corey Bardman's lab, I've been studying how nematodes, roundworms, right, as they're known, how nematodes adapt to environmental stresses and harsh environments. Um, and so, yeah, this summer I was able to pursue independent research into a novel nematode species, one that I helped to discover, that I hope will have a lot of environmental um, implications, environmental science implications. And I was able to do this at the Biological Laboratory at Woods Hole in Massachusetts. It was a tremendous experience, and yeah, I'd love to tell you all the stuff that I did there.
Awesome. Okay, so first things first, why do neuroscientists study worms? Why is that a thing?
Yeah, absolutely. So, one of the tremendously uh, convenient and useful things about worms is that, for instance, the laboratory nematode, Cenorhabditis elegans, has 302 neurons. So, numerically, very simple brain. That makes it very reasonable for us to be able to study their behaviors and the actions of their signal neurons and see how it relates to things that the entire organism is doing. Um, and the other convenient thing about uh, neurobiology with the nematodes is that their neurons pop up in sort of fixed positions from individual to individual. So we can even give those neurons names. We are able to know what those neurons do. For instance, this neuron senses salt. This neuron senses oxygen. This neuron propels the worm forward. This one moves it backward. So it's a high degree of control that is really um, uh, insightful and is what allows us to kind of leverage this simple animal that still has complex and quite beautiful behaviors.
Awesome. Yeah, so they have there's a lot of accessibility with them as a model system.
Right.
Okay, so going to your work, you were looking at arsenic tolerance. Can you give us a bit of background about why you were studying that, what it means for the general field, how those worms are different than the ones that you just described, the C. elegans?
Totally, totally. So I'll start with right now, the world is facing a global arsenic crisis. So that's kind of the larger context of all of this. 230 million humans worldwide are exposed to dangerous levels of arsenic, largely through groundwater and environmental pollution and environmental sources. And uh, arsenic is a extremely potent neurotoxin. It's able to damage the body and our nervous systems very effectively, and I can get into kind of how and why that is in a bit. But the fact of the matter is that we don't understand arsenic toxicity as well as we would like, and as a result, there are no available treatments or cures for arsenic toxicity. And the symptoms of arsenic toxicity can range from peripheral neuropathy, numbness, pain, mood disorders, cognitive disorders, and of course, death. And so it's something that we're extremely motivated to understand better. And I got into this because as part of my PhD towards the end, um, me and several postdocs in the lab, as well as a fellow graduate student, decided to go to Mono Lake in California, which is this beautiful and very famous lake right near Yosemite. And it's an incredible lake because you look at it and it kind of looks like what you imagine maybe Mars might look like in parts of it. It's incredibly desolate, and it looks very hostile. Um, it's an incredibly arsenic-rich lake amongst other things. And we went in looking for worms, and indeed, we were able to find several species of worms that can survive in this arsenic-rich environment. And from that, we were able to culture one of those species in the lab and continue to study them and compare them to C. elegans, which is not from an arsenic-rich environment normally. And by comparing the two, we hope to understand the strategies that this species from Mono Lake has for dealing with arsenic, which we hope may inform uh, treatments or therapies for humans, as well as just help us with some basic science questions of how do animals adapt to high arsenic levels, and how do they evolve to um, survive.
Okay, so my understanding is that you want to look at these worms because they are specialized for a certain thing. That makes a lot of sense, especially in a comparative context and comparing specifically their neural circuits of a C. elegans to one of this new species that you discovered. So, could you tell us a
Right.
little bit about kind of the experimental approaches that you take to look at the differences? And I know that you have some slides and some videos, and I'm sure the viewers would love to see those. Um,
Definitely.
so yeah, could you tell us a little bit about kind of the practicality of when you're doing this work? What does that look like?
Absolutely. So yeah, let's bring up some of those slides right now. Um, yeah, so this is an earlier slide of where I did the work, and this is a photo of us. This is the 2023 Grass Lab. So I did my work here with this fantastic cohort of fellows. As part of the Grass Laboratory, and it was such an inspiring place to work in, and I just want to give them a shout out before I kind of dive deep into um, the specific experiments that I did. And so my cohort, they worked on such a diversity of organisms, ranging from comb jellies and flies and squids and little skates and frogs. Um, so I was able to really get a lot out of this environment. And so moving on to the next slide. Oh yeah, this is sort of, we can uh, return to this in a second. But if we go to the next one, there we go. So the specific thing that I studied and I compared between C. elegans and T. tufae was that, oh, and I should explain, the worm that we isolated, the new species that I helped to discover back in my PhD, we named it Tocorhabditis tufae. It means the birthing nematode of the tufa towers. And that's in reference to the fact that we isolated these worms near these tufa towers in Mono Lake. So these are these famous structures in Mono Lake that are these salt towers that precipitate up and over the lake surface. And um, as you can tell from this photo here of the tufa towers coming out, the lake is very saline, very alkaline, very arsenic rich, as I had mentioned. And so when we took a look at T2Phi, the first thing that we immediately noticed is that it has a remarkable and rare birthing behavior amongst nematodes. So no, over 99% of nematodes, including C. elegans, they lay their progeny as eggs, whereas T2Phi, yes, here's a video of C. elegans laying an egg, and T2Phi, on the other hand, if we go to this next video, there we go, holds its eggs in its uterus, hatches them there, grows its progeny through several molts, before giving birth to well-developed larvae. And so we took a look at this, and immediately our first um, guess was that this gestation behavior might be a way for the mothers to protect their progeny from the harsh environment of the lake, including the arsenic. And so one of the experiments that I was able to complete in the grass lab this summer was these dissection experiments that you can see schematized on the right of the slide here. And I believe we have a video of what this dissection looks like. And so I take a needle, a 30 gauge needle, and I cut the heads of the mothers. And that's because their heads are just anterior to where the uterus of the animal ends. And so that clear tissue that you can see that's starting to protrude out from the animal is the uterus. It's being pushed out by the strong internal positive pressure of the animal. And the dark tissue that's being pushed out is the intestine. And I always like to point out in this video, the head of the mother is able to swim around on its own in this dissection buffer, which is, I think, very fascinating and macabre. And there we go. That's the moment that the uterus ruptures, and you can see how many larvae I can collect from a single dissection in this way.
So James, one quick question.
Of course.
How does the gestation period of T2Phi compare to C. elegans? Do you find that
Mhm.
they're really in the egg state for the same amount of time, and then the live birth, they kind of hatch in the mother at kind of a comparative time that the eggs would be laid in C. elegans?
Yeah, we haven't done precise timing of the egg development. Um, Although I have a collaborator in Japan, his name is Dr. Ryoji Shinya at Meiji University. He's doing more of those kinds of studies. Um, but what we find is that after the eggs hatch, normally the worms like C. elegans, they go through 4 larval stages before they completely mature into an adult. And in T2Phi, our observations are that it goes through 3 of those 4 larval stages completely inside the uterus before being born in the 3rd larval stage.
Okay.
Yes.
Okay, great. Okay, so how now does this live birth or this live birthing behavior connect to the arsenic tolerance that you were talking about earlier?
Absolutely. So this is why the dissection experiment was really useful for me. So what I did was I dissected out the larvae and I exposed them to arsenic earlier than they would have been if they were just naturally born. And I measured their survival in that condition. And then I compared their survival in those conditions to that of naturally born larvae that were also exposed to arsenic. And so if we go to the next slide, I'll be able to show you the results from that experiment. There we go. So if we see here in the green, that's the dissected larvae. And when the worms were dissected out from their mothers, they actually survived that procedure very well. On average, they survived 96% of the time. But if I expose those dissected larvae to arsenic, I see a drop off in survival, such that 1 in every 4 larvae on average died. Um, and this is in comparison to naturally born larvae survived just fine with or without arsenic. So this really indicated to me that gestation really does protect the larvae while they're developing from arsenic. And that once they are born in that 3rd larval stage, they've actually developed the resilience that they need in order to continue surviving from that point onwards. And so um, what I'd like to do is, of course, test the other harsh conditions of Mono Lake, such as the high salinity and the high pH, and see whether the gestation protects the larvae against those conditions as well. But another thing that I'm really excited about following up on right now is just how did this behavior evolve? Right? If we go back to those videos of the egg laying and the live birthing, we can see that the behaviors are very, well, they're dramatically different. But not only that, another analysis that I was able to do during my Catholic grass fellowship is if we let this video repeat over and over, is that so with egg laying and C. elegans, a lot of that is controlled by the mother. I mean, all of it is controlled by the mother. Her muscle movements and contractions are what's necessary to drive the release of the egg out from the uterus. But if we focus on this video here of how birthing works in T2Phi, we can see that it actually appears to be driven, at least in part, by the larvae. And so the larvae are always active at the moment of their birth. They're always birthed head first. They always come out head first. And they always uh, enter this really active period inside the uterus before they're born, where they coil up into this S-shaped pose that they seem to use to push themselves out. And you can see that S shape right here, where the worm has coiled and it pushes itself out. And so, what I'm really excited about following up on is just how did the control of birthing move from the mother, as you know, is the case in the ancestral behavior of egg laying, how did it move over to the larvae? You know, how do the larvae know when it's the right time to be born, and how do they drive their own birth?
Awesome. And just a quick question before we wrap up. So
Of
do we,
course.
so in C. elegans, we know the neural circuit that controls birthing behavior. And so my understanding
Yes.
now is that you're looking at that circuit in T2Phe to see any
Yes.
differences. So I'm curious if you find any differences in the circuit architecture or kind of the activity of neurons or kind of what we know about the differences in those circuits and those resiliences.
Absolutely. So that's a wonderful question, and um, one thing that I forgot to mention is that a convenient thing about worms is that they've got an incredibly constrained body plan, and they're incredibly constrained in the number and types of neurons that they have. And so I mentioned that C. elegans has 302 neurons, and related species have a similar number of neurons. And so that really allows us to compare between the neurons and the nervous systems of distantly related worms and draw those types of conclusions that you are talking about. So when I compare T2-Phi birthing to what's known about C. elegans birthing, immediately what I found is that birthing in T2-Phi is under distinct neuronal control. And so all of the neurotransmitters that are known to modulate C. elegans egg laying, and these include serotonin, acetylcholine, and tyramine, all of those do not have similar effects in T2-Phi. And in fact, there is a key serotonergic neuron in C. elegans that helps to drive egg laying. It's the major driver of the egg laying behavior. And when I look for it in T2-Phi using antibodies to stain for serotonin and to find where the serotonergic neurons are, I don't see that neuron in the T2-Phi birthing circuit. So somewhere in the evolution of this behavior, that neuron, or at least serotonergic expression from that neuron, has disappeared.
Hmm. Wow, that's so fascinating. A lot more to do, I'm sure. Well, James,
Absolutely.
thank you so much for joining us. I want to commend you on taking this observation from environmental change, anthropogenic change of arsenic through groundwater, and kind of connecting that perturbation all the way down to a single circuit within Tichofe. That's very fascinating and inspiring to me. So thank you very much, and let's follow up because I have a lot more questions.
Absolutely. I had a great time. Thank you very much, Angie.
Thanks, James. Talk to you later. Bye.
Bye.